You will hear a voice.
John, you're muted.
Sorry, thank you, Aruni, and thank you everybody for participating there. Shane tells me that we'll all be muted during the talk, and that if you want to speak at the end of the talk, then you have to unmute yourself, put your hand up and unmute yourself. But it gives me great pleasure to introduce our speaker tonight, Dr. Edmund Gilbert, whom we all know. And I'll have to read out what I have here about him, because it's all very highbrow to my way of looking at things. And he is a population geneticist based at the Royal College of Surgeons of Ireland, and he's currently a lecturer in computational genetics. And he completed his PhD in Irish population genetics at R.C.S.I. in 2019, researching the genetic history of Ireland and Scotland with the Irish DNA Atlas project. And this was an academic research collaboration with the GSI itself since 2011, as well as reporting an analysis of Irish traveller genetic history. His current research interests aim to reveal the demographic history of populations and specific genetic mutations through the use of haplotypes, which is the closely linked genetic markers that are handed down to us. And he, as well as extending our knowledge of Irish genetics to diasporic populations. I'm very sorry, Edmund, I'm reading from a very badly printed piece of paper here, but nevertheless, I don't want to take from the importance of the work that you do and the way you've cooperated with the Society. So, ladies and gentlemen, it gives me great pleasure to introduce Dr. Edmund Gilbert.
Thanks, John. Thank you. And thank you to the Society again for inviting us, RCSI and me, to talk to the Society again. I think it was just over a year since last I was able to present to the Society, so it's lovely to be back here. I'm going to try to share my screen. Try being the operative word. Hopefully you guys can see that. Okay, right. Yes, thank you again for the introduction. That was great. So today I'm going to be talking about what we've been using the Irish DNA Atlas to do to learn about these diasporic populations. So we're going to be looking at one population in general, in the Canadian province of Newfoundland and Labrador. First of all, what we're going to be doing is giving just a quick update on the Irish DNA Atlas itself as a project, and then for the bulk of tonight's talk, I'll be talking about the analysis that we've been doing as part of the Newfoundland and Labrador project. So very quickly for the Irish DNA Atlas, we've recently secured funding, extra funding from a research centre or funding body, well, not funding body, but project called the Centre for Research Training in Genomic Sciences. So this is an SFI, SFI standing for Science Foundation Ireland, which is a large funder of science research in Ireland. Is a centre for PhD students. So every year, about 20 PhD students are selected, and they do genomic or kind of computational biology projects. And we have a number of them working in the lab, including one who's working on the Irish DNA Atlas herself. And as part of this centre, they make available extra funds to generate data that are of use to Irish genetic science. Which is including basically allowing us to double the amount of genetic sequences that we have on file for the Irish DNA Atlas. So our SNP data that we have contains about 196 individuals from the Irish DNA Atlas, and they formed the core of our original paper back in 2017. And we're going to be doubling those sequences with new individuals that we've recruited since. So we should have about a sample size of about 400 by the end of that. So we're currently working on getting the DNA sequences and then, or the DNA samples rather, plating them out and then sending them off to get genetic data. So the work itself, using the Irish DNA Atlas, it's continuing to be worked with. We have a PhD student I mentioned before, someone called R.C.S.I Shanmugam, and she is working on extending the results that we worked on at the back in 2017 about the the population structure of Ireland, so different genetic groups in Ireland, but instead this time looking at the demographic history of those groups. Now we'll go into demographic history as a concept with the Newfoundland project, but hopefully we should get Ashwini to present to you guys hopefully by the end of this year. It's all coming together at this stage, which is quite a nice, exciting time for her. The second point to update is this Newfoundland Ancestry Project, which I'll be talking about for the rest of the talk, but I just wanted to quickly give an overview now. So this is in a collaboration with a genomics company based in Canada that's called Sequence Bioinformatics. So they came to us because we have expertise in population genetic analysis, but also that we have this access to very high quality Irish references with geographic data. Basically, this is the only data set of like this that exists. And of course, this is the Irish DNA Atlas. And so even the fact that the Sequence Bioinformatics sought us out is a nice demonstration of the value of the Irish DNA Atlas and what we've been able to accomplish as a collaboration. So this project is essentially looking at the European ancestry that is predominant in the population of Newfoundland and Labrador, specifically the European population, so the settled or the settlement population. This is currently at the moment unpublished work. I'll give a quick overview of the current publishing state. But at the moment, if I kindly ask you not to take any pictures or circulate them beforehand, hopefully within the next month or two, we should be able to go public with the actual paper that should be freely available, and so we can coordinate a nice release with this video and the paper as well. But for now, kindly no recordings, apart from obviously this recording that's going on right now. So let's give an overview of the projects. The project's entitled the Newfoundland and Labrador Mosaic Founder Population, descends from an Irish and British diaspora about 300 years ago. And hopefully during the course of this evening, I can convince you that this statement is true. So let's very quickly introduce Newfoundland and Labrador. So Newfoundland and Labrador, although we're mostly focusing on the island of Newfoundland, is a Canadian province in the Northern Atlantic. This is where it is on the map. You can see in red the province, which is on the mainland, as well as the island, which is part of the province, which is the island of Newfoundland here. It's a large proportion of the European settlers in the 18th and 19th centuries were English and Irish fishermen and families. And we know this because of genealogical records through genealogy societies in Newfoundland and associated historians. And from those records, it suggests that a lot of these English and Irish fishermen and families came over from ports that were from the southwest of England, Southeast Ireland. So one of the questions is, so were the individuals that were going on those boats in England and Ireland, were they from those regions, i.e. from the southeast of Ireland and the southwest of England, or were they from all over Ireland and England, but they just happened to get on the boat at those areas, the southeast of England and the southwest of England and the southeast of Ireland? Interestingly, Irish was spoken on the island until the very early 20th century. In certain places around Newfoundland, and indeed, if you listened to the accent in Newfoundland, it's quite familiar, at least to my English ears, to a number of different Irish accents as well. So there is this real case of that there's this strong Irish diasporic ancestry that's in Newfoundland and Labrador. This is a bit of a zoomed in map of Newfoundland, the island itself. You can see Labrador on the mainland up the top of this map. And so Newfoundland itself is a large island. We can broadly divide this into the northwest, the southwest, and the southeast. This whole peninsula here in brown, red, and yellow is the Avalon Peninsula. This is where the majority of the population in the province reside. So in all of the province that you see here on the mainland here, the majority of the population actually reside only on this small part of the island itself. And most of the communities are based around these bays around the coastline. the inside of the island is pretty inhospitable, or at least resistant to settlement. So what you'll find is that there are a load of outports, essentially, small coastal communities scattered around the island. So there are a number of different bays, such as Bonavista, Trinity, and Conception, as well as the peninsula of Burin down here. And these are a couple of names and a couple of geographies that we're going to be learning about later in the talk. The the provincial capital of Newfoundland and Labrador is St. John's, and this can be shown here on the top of the northern part of Avalon, of the Avalonian Peninsula. So previous work, there has been some genetics work performed investigating the genetics of Newfoundland. Genetic mapping, or the identification of genetic regions that are implicated in disease risk. So if there's an inherited health condition, and tracing the genetics of that health condition to a specific area of the genome has been relatively common through clinical work in Newfoundland and the doctors that reside in the province. And this is and because of this, as well as historical and obviously geographic information, the genetic isolation of Newfoundland has been appreciated for some time. And so a study back in 2016, R.C.S.I et al. Looking at the population genetics using a small data set of about 500 individuals with some SNP array data. And they found that there was for example, a split between individuals that were reporting as Catholic as background, which are these blue triangles, and then a Protestant in background, which are these red crosses. They also noted that there are individuals who they didn't designate Aboriginal, although we're going to be using the label Indigenous American for the rest of the talk, in these black circles as well. So you can see each one of these dots represents the genotype or the genetic identity for one individual. So you can see that individuals from the same religious denomination tend to cluster with each other. And then there are also these groups of Protestant individuals that seemingly fall out or are genetically distinct, shall we say, from the bulk of the individuals from Newfoundland and Labrador. This is what we call genetic structure or population structure, where individuals appear to form their own groups based on a shared genetic identity. So as you can see, we have this genetic gradient between Catholic and Protestant background, which is quite interesting. We know that the majority of Irish settlers were of Catholic background, and the majority of English settlers into Newfoundland were of Protestant background. So this might have something to do with that ancestry. And indeed, Zayatel found that there was evidence of this where they so now all of the Newfoundland individuals are shown as black crosses, but below them you can see that there are blue individuals who are British ancestry controls, and these red individuals which are Irish ancestry controls. And you can see what looks like quite an overlap of the British, but also some overlap between the Irish space as well. So it looks like there's this mix between British and Irish ancestry here. But this was essentially, and also that we also have an idea that Newfoundland is indeed isolated. Looking at these segments called ROHs, or runs of homozygoty, this is a kind of a measure of how related your parents are. And obviously small populations, parents tend to have an elevated background relatedness just because of a small population size. And we can see that Newfoundland, using these box plots, has an elevated level of these ROHs, meaning that the average parent of a Newfoundland individual is slightly more background related than the average British individual or the average Irish individual. So this is the limit of what we knew beforehand. And what we'll show today is that our population expertise, as well as the references that we have available thanks to cohorts like the Irish DNA Atlas, has allowed us to trace this back into far greater detail and really extend our understanding of the genetics of Newfoundland. So what we wanted to do was investigate the genetic landscape of Newfoundland and Labrador, essentially like what we've done in Ireland using the Irish DNA Atlas. What we also wanted to do was combine these Newfoundland individuals with our population references like the Irish DNA Atlas, as well as other British references, to investigate the presence of Irish and British ancestry in Newfoundland, using these valuable ancestry references, like these reference groups that we reported back in 2017. And what we can do then, hopefully, is then trace the Newfoundland ancestry back to Ireland and back to England into the regions where the Newfoundland European ancestors are from. So what we did was condense all of these questions, all of this context, into 3 addressable questions, 3 project aims that we wanted to have a look at. And you can see in bold, these are our kind of population genetic questions, which are a little bit jargony, as is often the case from a scientific community. So what we have at the end of the day are some more accessible questions. Are there geographic regions? How different are geographic regions within Newfoundland? How genetically different are communities that live in the same area within Newfoundland? Is there this question of population structure? The second question being, do these differences follow British or Irish ancestry? I.e., are there genetically different regions in Newfoundland and Labrador that are different because they share different proportions of English or British and Irish ancestry? And then finally, what we wanted to do was look at how isolated is the genetics of Newfoundland and Labrador. It's out in the western coast of or the northwestern coast of the Atlantic. It's pretty hard to get to on a small population size. So what's the evidence that context has shaped the genetics of Newfoundland? So what we're going to do is split the rest of this talk up into these 3 different sections and hopefully show you that the analysis that we were able to do in conjunction with the Irish DNA Atlas was able to answer these questions. So very quickly, I wanted to quickly do a quick current status before we get to those results sections. So yes, the project is now complete. We've done hopefully our final analysis that was completed last Friday in a hurried analysis that I was completed towards the end of the year, towards the end of the week. And what we've done is resubmitted our final finished manuscript to the journal. So we're waiting on final acceptance for the journal. We've gone through peer review, and the manuscript has really improved, given a number of different rounds of communication with our peer reviewers. So hopefully in the next month or 2 we'll be able to go public after the journal publication. Hence no recording or pictures of it or screen grabs tonight. But hopefully this recorded lecture itself will be able to go public along with the journal publication to really show off the results. So the headline finding for the paper that you'll be able to see hopefully in a month is that we've shown the role of regional Irish genetics in the European settler history in Newfoundland and Labrador. And to do so hasn't been possible without using cohorts like the Irish DNA Atlas. The Irish DNA Atlas is truly a unique resource that allows us to perform this research. So what we found out is that Newfoundland's communities are connected by water. Usually when we look at the population structure within different communities, these are groups that are grouped by different geographic regions. And instead, what we find is that Newfoundland's communities are actually connected by water instead. Newfoundland's English and Irish settlers are echoed in the current landscape, so we can still see marks of that differential settler of different English and then different Irish settlers still found in that genetic landscape of Newfoundland today. And we've also found substantial evidence that Newfoundland can be considered a founder population that had this founding effect around 300 years ago. So let's have a look at community structure first. So what we did was perform clustering analysis on about 1,800 Newfoundland individuals from this project that Sequence Bio is running called the Newfoundland and Labrador Genome Project. And as part of that project, we have information, kind of initial information on whether genetically they appear to be mostly of European ancestry, or whether they include an ancestry component that is looking like it's indigenous American ancestry. So we have some initial information on whether an individual is of European and indigenous American ancestry. Of course, at this point, we didn't know what type of European ancestry it was. So what we're doing is performing this clustering to identify genetic communities, i.e. individuals who form groups that share recent ancestry. And what we can do using these clusters is to look at different groups within Newfoundland with a greater resolution than what we were able to if we were just looking at Newfoundland on its own. So when we group those 1,800 individuals together, and cluster them using this program called Find Structure, what we find is 22 such clusters emerge. We can see the cluster names here with the number of individuals in each of the clusters, and you can see that there are quite a lot, a few very large clusters, and also a number of different very small clusters as well. And the names that we assign these clusters are reflective of the different areas of Newfoundland where the majority of their grandparents come from, in a similar way that we did in the Irish DNA Atlas paper. And we can see that these clusters are themselves grouped together on branches of a dendrogram. So we can see that these clusters with individuals with grandparents from the Avalon Peninsula do tend to group together as well. And we have this south coast that's related to the Burin Peninsula. We also have a number of different clusters in Bonavista Bay, as well as clusters within Trinity and the Conception Bays as well. We also have this interesting group of four clusters of individuals who together showed various proportions of indigenous ancestry. So what this bar plot shows is the proportion of individuals rather than the proportion of ancestry. The proportion of individuals that have were assigned to one of two labels, either being of European ancestry or being of indigenous ancestry. And you can see that here in this green and this orange color here. We can see that the smallest cluster, this Admix European Indigenous 1 cluster, actually all of these individuals were previously assigned to this label of indigenous ancestry. And we have smaller fractions for the other clusters that are grouped there. If we look at religious backgrounds, so these are self-reported religious backgrounds for each of the participants that have been grouped into these 22 such clusters, we can see this really striking divide between Catholic background and in red and Protestant background in various shades of blue, with any other grouping in grey. And you can see the individuals are grouped into this Avalon clusters and also this Burin cluster as well, are predominantly Catholic in background, whereas the majority of the other clusters around Newfoundland are mostly of Protestant or some sort of denomination of Protestant Christian. So either Anglican or United, where United is a Canadian denomination. So what we found is there are 18 clusters of what seems to be European ancestry and 4 clusters of indigenous ancestry as well. We can show that in greater detail using ancestry references. So what this is a plot of is the genetics of a number of different worldwide references. Now, an individual is represented by a stacked bar plot. So each one of these lines that goes upwards represents one individual, and the proportion of each of the colors on this line, so this individual here is about 50% this yellow line, and then 50% of red. And that's the proportion of their ancestry that is assigned to these 5 different, what we call components, but you can think of them as ancestry backgrounds. If we group these individuals by the same population that they were recruited from, and then we group these populations by world regions, we can see patterns that emerge. So all of these red populations, or the populations that are carrying a lot of this red component, are of African origin. So this is tagging African ancestry, this red component. You can see this African ancestry is also present in some of these American populations, these 4 American populations here, but also in our Spanish individuals down here at very small levels as well. This blue component, of course, is then an indigenous American ancestry component. It's highest in these individuals from Lima. In green, we have East Asian ancestry. And then in yellow, we have this European ancestry that's mostly predominant. So we can see there are slight differences. For example, Finland showing East Asian ancestry, and that's reflective of Central Asian, Siberian genetic connections that we're aware of as population geneticists. And then we also have in this purple, this South Asian ancestry component. So this is kind of the worldwide map of just continental ancestry groups. So if we project these Newfoundland individuals on them, and we group them by clusters, we can see that for the vast majority of our clusters, all 18 of our European ancestry clusters, they do indeed look European, European ancestry. Interestingly, though, our indigenous American individuals do show this detectable level of indigenous American ancestry, shown by this blue proportion. So these individuals do hold or contain this Indigenous American ancestry. Now, we were interested in European ancestry because as a study, we weren't designed or set up to be able to analyze Indigenous American ancestry in a safe and beneficial way to that community. So what we did was note that we had identified these individuals, but the study goals themselves were designed at the beginning to look at European ancestry. So what we did was focus on these 18 clusters moving forward. If we look at these populations, or these 1,800 individuals on a map, and we color code them by their religious denomination, we can see that there's some striking differences. So this is a PCA, or a plot of the genetic dimensions of our data set, where each dot represents the genetic identity of one individual in our Newfoundland data set. And we've color-coded them red being Catholic and blue being from a Protestant background. And you can really see that there is a genetic gradient between these two communities. Of course, it's not a 100% divide. You've got individuals that are who identify as Protestant, but they plot onto this this Catholic or the Catholic majority genetic space as well. So it's not a 100% dividing variable, but there is this pattern in the data. If we look at the clusters themselves, so if we bring back our dendrogram or a tree of these 22 clusters, and then we plot those individuals on the same genetic dimensions, but we color code them according to these clusters, you can really see where these clusters are coming from. So our Catholic individuals are mostly from the south coast in blue, or the Avalon Peninsula in various shades of purple, and then our Protestant individuals are mostly from these Conception Bays or Bonavista or Trinity Bay clusters as well. So we can define these the Newfoundland European genetic structure along three main gradients, separating the Avalon Peninsula up in the top left here, Trinity and Conception Bays at the top right, and then down along the south coast towards the bottom of this plot. If we plot the grandparents of these individuals and color code those grandparental birth locations by the clusters that their grandchildren were assigned to in the present day, we can really see this community structure arise. Now, we've added a little bit of a jitter to each of these points, so which is why you've got some individuals that are actually that were born in the sea, for example, over here, but that's just to preserve their anonymity. What one can see is where all the different communities are from around the coast in around the island of Newfoundland, because you can see these kind of grouped clusters where they are being placed in the same township. But you can also see that these clusters really are picking out different genetic regions around Newfoundland. For example, the Avalon South cluster in this rich purple color here. It's mostly found in this small community down in the south of Avalon down here. We can also zoom into Trinity and Conception Bays here to really show the remarkable structure along the coastline in these communities. Even to the point where on the western coast of Trinity Bay, we have this dividing line essentially down here between these communities and these genetic communities. So it's really quite amazing the level of structure or geographic structuring that we find in Newfoundland. We can zoom into individual clusters. So the mainland or the land is shown in a dark color with the sea in white, and you can see the individuals are placed around these communities. There is some spread, of course. People are connected across these waters, but there is trends within the data where certain areas of the island are where these communities are residing from. We can also see this in our green Trinity and Conception Bay clusters as well, where some of the structure is a little bit more is a little stronger as well. So we all can also see and show these this sharing network between clusters. So each one of these dots represents a space or what represents the value of one of these 22 clusters, and we can record the connections between these clusters in a graph like this. So what we can see here is that the various different regions around Newfoundland and Labrador seem to pop out. We can see these connections quite visually. Now, this network here was specifically chosen to be the most minimal connected network available. So it does look like on this graph that, for example, the Avalon Peninsulas clusters are not connected at all to the rest of Newfoundland. That is not the case. What we did was choose the lowest number of connections that would connect all of our clusters to another cluster. And basically it shows that on average, or relatively speaking, our Avalon clusters, or at least these two Avalon clusters, are quite disconnected compared to the rest of Newfoundland, which makes sense because these Avalon clusters are as we'll see, of a different sort of ancestry background to the rest of a lot of the other clusters. And what you're seeing with the length or the width of these lines or connections between these clusters is that the thicker the line, the more genetic sharing between the average individual of these clusters. So we can see, for example, the South Coast pop out as well, where we've got these Burin east clusters that are grouped together, but also have connections to another south or west coast here. So one question that came up in our review at the journal was, are these clusters separated by geography or religion? One concept within population genetics is isolation by distance. Basically, the fact that individuals tend to have children with other individuals who are geographically proximate, i.e. you choose your partner who's close to you for a lot of human history. And so when you see clusters across geography, like you do in Ireland, the easiest explanation for those clusters being shown in being ordered by the geography is that, of course, individuals tend to share genetic identity with individuals who are geographically close to them. But we argued that there is some evidence that some of this clustering is down to religious behavior rather than just geographic distance. So our reviewers asked us to go into this in a little bit more detail. So what we did was look at whether clusters from same or different denominations, i.e. if they're clusters of two Catholic clusters or two Protestant majority clusters, are more genetically distant to each other than expected. So this is what we see here on this graph. So each dot here represents the total or the average amount of genetic sharing that a pair of these Newfoundland clusters share with each other along the y-axis. So, and the colors indicate the type of pair. So purple is between a Protestant or Protestant cluster pairs, so between two Protestant clusters. Green between two Catholic clusters, and then orange being between a Protestant and Catholic pair, so across the religious boundary, as it were. And then on the x-axis is the median geographic distance. So you can see that as the distance grows closer, the amount of sharing goes higher between all of our different types of clusters, and that's to be expected, i.e. people will tend to share more of their genetic identity with people that are closest to them. But what we see is that our clusters, on average, that between clusters that share the same denomination do share a lot more than expected compared to a pair of clusters that are across the religious divide, for example. So this shows that there is some evidence to suggest that religion is driving some of this genetic differentiation within Newfoundland and Labrador, this difference between a Catholic background and a Protestant background. So moving on from community structure within Newfoundland, we then went on to look at the exciting, at least from our perspective, question of what kind of British and Irish ancestry is there within the European settled community in Newfoundland. So to do this, we utilize what are known as identity by descent segments. These are commonly used within genetic genealogy, so I'm sure that a number of you are familiar, but as a quick recap for those that are less familiar with IBD segments, what these are essentially sections of the genome that are shared between two individuals. So we have a nice diagram shown here, where we have these two pairs of individuals, two pairs of individuals who share the same section of their genome. That is this orange segment here, and the upper and lower bounds of that section of the genome that they share in identity is shown by these horizontal lines. And that is because they are both descendants from this ancestor that was this orange, that held one single copy of the genome that's in orange here. And you can see that that copy of the genome gets recombined with meiosis with their partner to create children. Or then hybrids between the paternal and maternal chromosomes of each of the father. So you can see that this is now a mixture between the blue and orange between these two children, and then eventually these children's children have these IBD segments. So this IBD segment between these descendants are essentially tagging this common ancestor. Now, there's a number of different trends in these IBD segments. One of them is that the older the relative, i.e. the more distant in their family tree the relative is, what tends to happen is the smaller this IBD segment tends to be, because as we can see, recombination, which is this process where the haplotypes from the mother and father get recombined or shuffled together every in the generation. Recombination will progressively make sure that these segments become smaller and smaller as it shuffles the deck, essentially. But so in genetic genealogy, these are really useful for detecting close relatives or semi-close relatives, as well as being able to estimate the kind of the recent community that you're from. And indeed, in population genetics, they're very useful for that as well. So what we can do is look at whether the rates that these communities share these IBD segments, because these IBD segments are tagging a shared ancestor. So therefore, related populations or populations that share some ancestry or recent ancestry should share a lot more of these IBD segments than more unrelated populations. So this is what we did for our Irish, British, and Newfoundland populations together. So what we did was record the amount of sharing of these IBD segments between our Newfoundland clusters. So I'm taking the average essentially between these Newfoundland clusters, as we can see here. Along the diagonal, we can see the amount of sharing within each of the populations. And then what we did was also add in and record the sharing within our British populations, and then our Irish populations over here. See if I can add a pointer. No, I can't. Okay, hopefully you can see my arrow, but between Ireland. And what we can see is there's not a lot of sharing outside of these populations, because these the populations tend to share more within their own groups. So we can see Wales, for example, these three Welsh clusters in the middle here share a little bit. So you can see this darker, more kind of hotter color over here. But what we can see within Newfoundland, for example, is there's a much more elevated level of sharing, just generally speaking, across the province than we see across all of Britain or around Ireland as well. But you can see that Ireland, for example, is a warmer colour than Britain, mostly just because all these Irish clusters are a little more closely related to each other than the average British cluster between, Wales or England or Scotland, for example. But what we can see is that there is this hint of sharing between our Avalonian clusters. So this here, this slightly darker color here.
No.
Between these darker colors here, showing that there might be this Irish connection between specifically the Avalon clusters over here. So what we did was look into this more detail. So these are our clusters that we actually looked at within Britain and Ireland using kind of an updated reference set, also including the Irish DNA Atlas. So what we wanted to do is identify whether these specific communities, as we denote here, can trace back to Newfoundland and Labrador, or which ones kind of overshare their genetics with the European communities within Newfoundland and Labrador. Interesting, what we also were able to do was split apart Central South England into a number of different clusters, including East Anglia, Northern England up here, of kind of the Midlands up here, as well as South and West England in this dark red in circles, as well as separating Cornwall away from that as well. And then we can also see that we've got some nice dividing between the south of Leinster within the north of Ireland, as well as Munster and the south of Ireland over here as well, kind of recapitulating what we found in our Atlas paper. So what we did essentially was look at trying to model Newfoundland, or the European clusters of Newfoundland, as a mixture of all of these Irish and British groups. If we sum up their contributions between our Irish and British groups into each of the 18 European ancestry groups, we find this sort of pattern. So this is summing up the average ancestry proportion. In red is Central South England, so essentially most of England in red. And then in blue, green, and purple, various different groups within Ireland. So Northwest Ireland, Southeast Ireland, and Southwest Ireland. And you can really see that there's this structure, there's this differentiation between clusters that are mostly of English ancestry and these clusters that are mostly of Irish ancestry. And specifically, the Irish ancestry that it's being picked up from is from the southwest of Ireland and the southeast of Ireland as well. Interestingly, these are from the Avalon Peninsula, but also there is one particular cluster that keeps popping out along the Burin Peninsula over here as well. If we look at the specific areas around Britain and Ireland that is contributing to the signal, we do find that the English signal for this ancestry is being driven largely by Cornwall, Devon, and the south and west of England. So exactly where these boats were coming from towards Newfoundland. In the Irish context, what we can see is that these clusters are being driven by the southeast Leinster, as well as southwest Ireland and northwest Munster clusters, mostly. And what's interesting is that it looks to be the same sort of signal, but just different total proportions. So within our English, in inverted commas, ancestry Newfoundland clusters, is this similar sort of proportion of Cornish and Southwest English ancestry across all of these clusters, and the same within Ireland. It's mostly the same kind of ratios of all of these different clusters, which would suggest that they're from the same sort of origin. There's not different types of English ancestry or different types of English migration into it. It seems that the source of this English ancestry is from a specific unified community across all of Newfoundland, and the same with the Irish ancestry itself as well. We can test this with an alternative method. So that method was looked at counting up these IBD segments that are shared between Britain and Ireland. But we can also look at another method that's using this test statistic called Patterson's D. We don't need to go into this too much detail, but essentially what we can do is set up the test that we can test this. We can record the estimate of this D statistic given an outgroup. So these WRI stands for Eurobans. Which are an African population, sub-Saharan African population. NL stands for Newfoundland and Labrador. So each one of these y-axis clusters. And then we can test the sharing between Newfoundland and Labrador between England and Ireland. What this means essentially, if a cluster projects towards the right, they have more of an Irish affinity than the clusters that project to the left, which have more of an English affinity. And you can see that this signal of one of the Burin clusters and all three of our Avalon clusters is recapitulated and agrees with those IBD segment-based estimates. Interestingly, if we bring back that denomination data, which is shown on the x-axis here, so frequency of Catholic background in each of the clusters, and then we model set a trend line through that with the y-axis being the actual Irish ancestry or the genetic Irish ancestry proportion that we estimate. You can see there's this really nice relationship between self-reported Catholic background and Irish ancestry. The R squared of this is something like 0.8, 0.9, and it's quite significant. So that's the Irish and British ancestry that's in Newfoundland and Labrador. So we can move on now to demographic history. So answering the question, how isolated is Newfoundland and Labrador compared to Britain and Ireland? So what we did to answer this question is, again, utilize these IBD segments in quite a lot of detail. So what I want to do is very quickly go over the effects of different population histories or demographic histories, and how that impacts the sharing between pairs of individuals from the same community or same population. Because this will hopefully make the results in a couple of slides make a little bit more sense. So what we can do is talk about population size. So how many breeding individuals are in your population? And that can impact the kind of spectrum of IBD segments that you detect between individuals within that community. So what we're doing is looking at the IBD segments, or these segments of the genome that are shared between individuals who are from the same community. So if we have a large population and we have a small population to compare them to, if we look at segment length between the big population, we have this kind of this average segment length. So there's the segment length which is expected given the population history. But compared to, if we compare the large and small populations, the number of segments is different. So a big population size tends to have a fewer number of segments shared between the average pair of individuals from that community. And that's because they have fewer common ancestors on average, because there's a large gene pool. But if we take the small population size, because of everyone tends to be a little bit more related to each other, it being a small gene pool, to use the colloquial term, everyone shares a lot more common ancestors. And so what you do see is that there's a lot more of these IBD segments that are shared between the average individual pair, because people are, on average, more related to each other. So that's population size, but what we can also look at is the time period that this population size is happening in. So if we have a small population, or if we have it looking at a community, and they had a small population a very long time ago, we can compare and contrast that to the profile of a population that had a very small population very recently. We see there's a little bit of a difference. So we can look at segment length. So segment length is going to be lower, or lower on average on a small population long ago, where we have this increase of IBD number of segments. But to contrast with that, if we have the isolation very recently, the segment length is higher on average. The average segment is going to be higher because those segments are more recent, i.e. those common ancestors are more recent in the family tree of individuals in the same community. So what this means is that when we're looking at isolated populations, we can detect this small population size and give it some sort of time series or time information, and we can tag that with some time information. So we can see whether a population has been isolated a long time ago, or whether it's only been very recently isolated. So given all of these ideas together, what we can do is look at the demographic history of Newfoundland and compare and contrast it to Britain and Ireland. So this is what we do. So to use as a baseline, we can look at Britain and Ireland. So along the x-axis, what we're doing is like looking at the total length of sharing. So this is the total amount of the genome that individuals within that community share with them with another individual from the same community on average. And then on the y-axis, we can see the total number of IBD segments that are shared. So these have obviously quite a linear relationship, although not a perfectly linear relationship, as we'll see. So these populations here, these three groups are actually our Welsh clusters and our Acadian clusters. So we know that Orkney is a well-known isolate within Britain, and Wales, it seems also has quite a small, quite an elevated level of haplotype sharing compared to, say, England or Scotland in general. And these are our British or Irish references. In these grey dots, as we can see, are actually the results of our Newfoundland analysis. So let's look at those in a little bit more detail. So if we show here, so these coloured dots are now representing the average amount of sharing that is within each of these fine structure clusters or specific communities within Newfoundland and Labrador. The grey circles here, highlighted in blue, are the average for the British Isles and Ireland. And then these red are kind of the outliers of Britain and Ireland. So these are the communities that we would consider to be relatively isolated in the context of Britain and Ireland. And what we can see is that the average within Newfoundland and Labrador is really elevated compared to the average that we'd see within the rest of Ireland or the British Isles. We have these blue on purple clusters that tend to be the Avalon or the south coast of Newfoundland. And indeed, we have the highest amounts that were shared between individuals within Trinity and Conception Bay. So it looks like these communities are specifically more isolated than others around from Newfoundland. So even within Newfoundland, there is this difference of demographic histories where some communities appear to be more isolated than others. So we covered that a small population long ago tends to be associated with a lot of IBD segments that are short. Now, we can give this, we can look into this relationship between population size and time ago. We can look at this relationship and be able to build models to estimate the actual population size from IBD segments. So we can do that within Britain and Ireland. So this is what happens when we run a program called IBD-NE, which calculates the effective population size, or we can call it population size for the purposes of this talk, across Britain and Ireland. So this is over time, a time ago in generations. So this is present day in 0, and then going back 50 generations, where a generation is about 30 years. And then along the y-axis for both of these is population size in a log 10 scale. And we have this horizontal black line at the same place on the y-axis, just to be able to show the comparatively Britain and Ireland. And what you can see is that there is some more significant differences between our British clusters over here in various different, or British regions at least, within Britain. And then within Ireland, we see that's actually a far more stable, or the groups or regions within Ireland tend to have a more of a consistent population size, which seems to suggest that Ireland has had a pretty consistent demographic history across the island of Ireland. Although we're using and calculating this data for Britain and Ireland to compare and contrast to Newfoundland. So what we're actually doing is a kind of a preview, or a preview to what Ashwini's work is. What she's doing is extending this this analysis into a lot more detail, using a lot more different techniques to really drill into this data in a lot more resolution. So hopefully you'll be able to see that soon. But anyway, so what we've got is this estimates for Britain and Ireland, and we can see that they're showing around this 10 to the 5 region, and you can see the shape of the population sizes. They're pretty consistent, and they start increasing into the modern era around 10 generations ago. So what does this data look like if we look at the Newfoundland clusters on their own? It's pretty distinct. So these are our clusters within Newfoundland and Labrador. What we looked at is a small number of them, not all 18 clusters. We looked at the clusters where we had a large enough amount of individuals in each of the clusters to be able to estimate these accurately. And you can see that there's this substantial drop in population size around 10 to 15 generations ago in Newfoundland and Labrador. So what we're doing is tracing the number of ancestors that this individual had, or the population that those individuals belong to have. And you can see that there's this bottleneck effect, or this large contraction in historical population size around 10 to 15 generations ago. We see these large estimates of population size, but the confidence intervals around these are extremely wide. So these estimates, especially behind the bottleneck, are very inaccurate, mostly because a bottleneck tends to obscure the demographic history of a population behind it. Essentially, it's like wiping the board or scribbling over a whiteboard. It kind of obscures what was there before. But what we can see is this really strong evidence that indeed there was this contraction of population size, this reduction in common ancestors about 10 to 15 generations ago in the Europeans, or individuals with European ancestry in Newfoundland and Labrador. And so this roughly corresponds to about 300 years ago. This is hence 300 years in the title of this analysis. So we can also look at these ROHs, or runs of homozygosity, that we talked briefly about, given the existing work that had been done before we had run this analysis. So these show Newfoundland shows higher levels of these ROHs, which are a related concept to IBD segments, but are capturing information about parental relatedness. And at the end of the day, a kind of news headline part of this results is that Newfoundland also shows high levels of longer ROH and ROH in general compared to Britain and Ireland, as we can see here on this plot. So these circles show the kind of the average amount of all of ROH in Newfoundland and Labrador clusters, with the lines showing confidence interval sizes compared to Britain, and then also compared to Ireland. You can see the average is far higher than in Britain and Ireland. Even the areas of Britain that are known to be isolated, like Orkney or Wales, we're seeing a much higher degree of relatedness between the average pairs of individuals and pairs of parents within Newfoundland and Labrador. And this is consistent with a recent bottleneck within the population's history. So the last thing that we wanted to look at as part of this paper was asking the question, but does Newfoundland and Labrador share private drift? So it's all well and good to say and to show that Newfoundland is as a result of a mixture between Irish haplotypes and English haplotypes, specifically haplotypes from the southeast of Ireland and the southwest of England. But Newfoundland has also been isolated on its own subsequent to this founding, or this settlement that was driven in the late 17 and early 1800s. So Newfoundland has been isolated since that. So presumably Newfoundland has developed its own genetic identity in isolation using a concept called genetic drift. So this is why we're calling this concept private drift. So to do that, what we did was test each of the Newfoundland clusters using two different statistics, so which are shown on the x-axis and the y-axis. So each one of these dots here represents the value of each of those two test statistics that we performed, which is a Patterson's D test statistic. Kind of similar to what we did was to show the affinity to England or to Ireland for each of the Newfoundland clusters. But what we did here instead was test on the x-axis whether each of these clusters share an affinity to Irish or to an affinity with other Newfoundland individuals. So what we did was take a random sample of Newfoundland individuals for each of these clusters and test whether that cluster shares more genetic identity with these Irish references that we have, or these other Newfoundland individuals. So that's on the x-axis. And then on the y-axis, we did the same, but with English affinity compared to Newfoundland affinity. And what we get is this really interesting shape where we have some of these clusters that project upwards towards the right. For example, these green clusters. And these are clusters that share a lot of Newfoundland affinity on its own. They have a bias towards Newfoundland affinity when you compare them to English individuals, and they have a bias towards English Newfoundland ancestry when it comes to Irish as well. So this is reflective of this private drift of these isolated or more isolated Newfoundland regions. And indeed, these clusters are the ones that that pop out when we look at the degrees of isolation within our Newfoundland clusters as well. And then we have this other shape where these clusters, these clusters project away upwards towards here, where we see these Avalon clusters, i.e. these Irish ancestry clusters, tend to favor Irish affinity over Newfoundland specific affinity. But when you plot them according to English and Newfoundland affinity, there is still this evidence of shared private Newfoundland genetic identity compared to English identity. So that was the last of the data that we shared. So just very quickly to recap, and then we can I'm really happy to answer any questions or clarify anything. What we found is that community structure within Newfoundland and Labrador is strong and is organized along the coast rather than through the geography of the landscape itself. So this really leads on to the next question that would be extremely cool and interesting to look at, is whether that there are health-related genetic variation that also follows this regional differences. I.e., is there specific genetic conditions that are unique or more frequent in these genetic communities than other genetic communities within Newfoundland? I.e., can we tailor the genetics to understand disease risk? We've also found that Irish and English ancestry forms the European ancestry, and that Irish and English ancestry is still structured today. So we do see that there are regions around around Newfoundland itself that are predominantly Irish, that are predominantly English. So this obviously leads into the question of the genetic health in Newfoundland. Is that a subset of Irish and English genetic health? Are the genetic variants that lead to genetic health conditions in Britain and Ireland, are those present in Newfoundland? Because Newfoundland is a diasporic community of Irish and English ancestry. And then finally, what we've done is shown that Newfoundland and Labrador exhibits very strong evidence of a founder event. So this founder event may have influenced the overall risk of genetic health conditions, these disease variants that can lead to human health and human health disease. So has genetic variation to do with that changed because of this founder effect? These are all kind of future questions that this work naturally leads into. So thank you all very much for listening. I'd like to specifically thank especially the study participants, both from the Newfoundland and Labrador Genome Project, but also from the Irish DNA Atlas, the Trinity student cohort, as well as the people of the British Isles cohort, everyone that's provided DNA for these studies. Otherwise, we wouldn't be able to do it. To Michael Phillips at Sequence Bio, as well as the rest of everyone in Sequence Bio, including Alison Symington, who's helped with the manuscript. To Jean-Pierre Cavallari, who's the PI for the Irish DNA Atlas, as well as specifically Seamus and Michael, as well as the Genealogical Society in total for the Irish DNA Atlas. Part of this work was funded through my my fellowship. There's funded by the National University of Ireland. Of course, some of this work was also funded, or my time was funded by the Future Neuro Research Centre. And the Irish DNA Atlas itself was funded by Science Foundation Ireland. So I'd be more than happy to answer any questions. And that's my email address if you wanted to contact me afterwards. So thank you.